This session will focus largely on real-world evidence as a wraparound to your value strategy and talk through three different phases of it: early development, pre-launch, and then post-launch, and gain some perspective from our panelists. I'll do a little bit of cue up and tee up of the topic. My name is Eric Faulkner. I'm a Vice President for Evidera Precision and Transformative Medicine and have been working in the cell and gene therapy space for 25 years, back in the day at the bench when it didn't work until today when it does. But welcome, and we'll jump right in. I'll let each of the panelists go down the line and introduce themselves very quickly because they know themselves better than I know them. So I'll go ahead and start with you, Jane, and then we'll jump right in.
Great. Good morning, everybody. Thanks for getting up so early. So I'm Jane Barlow. I'm Senior Advisor to MIT Focus, their work on reimbursement for gene therapies. I also am Chief Clinical Officer at Real Endpoints.
Great. Good morning. Francesca Cook. I oversee pricing and market access for Regenix Bio. Regenix Bio is a gene therapy company with 4 products in clinical development. We have both products in the ultra-rare space as well as larger, more prevalent diseases in the retinal area. Looking forward to talking about at least how we approach from an early stage perspective real-world evidence.
Hi, good morning, everyone. My name is Bash Parasuraman. I'm the Vice President of Global Health Economics, Outcomes Research, and Market Access for Rare Disease at Pfizer. Pfizer Rare Disease, and our Rare Disease business unit was formed in about 3 years ago, and we have products which are older products in the inline space, such as in hemophilia and endocrine, and we have a very rich portfolio of assets in early clinical development to late stage development. A lot of them are gene therapy assets. Thank you.
Hi, I'm Ramesh Arjunji from Avexis, a Novartis company. I'm responsible for global health outcomes and real world evidence. So I support Zolgensma across all the indications, type 1, type 2, type 3, pre-symptomatic. And so I also develop global cost effectiveness models, budget impact models, and essentially generate the evidence that is needed to support the value proposition for Zolgensma. I also work on pipeline products, including Rett, ALS, and Friedreich's ataxia. Glad to be here.
Okay, thank you so much. And we'll go ahead and just set the stage a little bit here. Just definitionally, we're going to take a fairly broad view of how people would characterize real-world evidence because I think it will come into the discussion a bit. But in general, the FDA has a couple of top-line definitions: data relating to patient health status and/or the delivery of care routinely collected from a variety of sources. And variety of sources present certain challenges. Or clinical evidence about the usage and potential benefits and risk of a medical product derived from analysis of real-world data. So that's a very broad definition. Well, what does that technically mean? Where does this information come from? It comes from a variety of sources, including medical records, charts, electronic health records, claims data. It can be provider surveys. It can be disease or product registries. It can be patient-generated, patient-centric data in some cases. Historically, before all of these were baked into some of the thinking at FDA, we also had scenarios where you would have a literature review or meta-analysis, a statistical analysis of published data. So a lot of this basically is retrospective data. Some of it is prospective data. The choice of which you use here really kind of depends on several factors, including the development scenario, how crowded the space is, whether or not it's ethical to do a randomized controlled trial, and a number of factors. So where you pick your type of real-world evidence can vary, but all of these have been used in one way or another to support product uptake, and it's becoming increasingly important, as we'll see. So I won't go through this slide in detail. Real-world evidence is starting the journey that you saw with patient-reported outcomes some number of years ago, where we're stepping through, being driven by regulators from more of a nice-to-know towards maybe a got-to-have, but definitely towards having a value of use. And this just shows, the historical trajectory of where we're going right now. We're in about year 3.5 or 4 of a 5-year process where the FDA is having a bit of a think about how will this information be used, what will it be used for, what does good look like. They're still making up their mind. They're very open to engaging with manufacturers. And in general, from what we've seen looking across multiple case examples in this space, where you run into trouble is when you don't engage with the agencies if you're going to use real-world evidence, because they're trying to understand how this information is used as clinical trial models change. 50% of all the oncology products today and 50% of the rare disease products will launch or have been launching with single-arm studies, for which real-world evidence is kind of got to have to be able to show basically the comparative benefit. Then just another, this is the same kind of trajectory in the EU. That they're moving towards this same, a bit more slowly, albeit versus the United States in this case, but they are moving in this direction. How do we use data that come from unconventional sources outside of randomized controlled trials? Why do we care? Here are some examples, and I won't go through this, but you can see that there's a growth curve here that's happening in terms of use of real-world evidence in oncology product, non-RCT-based decisions. So this information is already, and has for some time, been used. Just because FDA is putting out a guidance does not mean that companies have not been using this for a long time. If you look back in oncology, as far back as Xalkori with Pfizer as one of the first single-arm launches that we've seen in the space, then you have to have that comparative data. But figuring out how to get there is sometimes challenging. Where is it used today? Just briefly, it's used all across the life cycle. It's used for regulatory designation purposes, you see here, but for approval and post-approval, for label claims and expansions, in the HTA process, although some health technology assessment agencies, particularly in Europe, are a little slower to come to recognize and incorporate this information versus the United States, for example, and then also used in constructing or supporting value-based payment. And we can talk about some of these things, but just to set the stage. I won't go through these examples. Here are some in oncology, but just some examples of how it's been used, historical controls, supplement to the RCT to broaden the label, comparative effectiveness, clinical trial reanalysis, certain of the methods, showing the data used here, and again, won't go through it, but it was used to support registration or accelerate the registration process to a certain extent. It's used to help color in the vision of what's happening with these patient populations in some cases in a way that you, in some cases, simply cannot get from RCTs. Here are some examples from rare disease and other advanced therapy examples. Many of these will be very familiar. Just looking at leveraging a registry to make best duration of effectiveness case with Strimvelis, which is one of the exemplars that still out there, coming to market with 9 or 10 years worth of data, which is challenging to do in most scenarios, but it was very powerful for them even with small populations. With Kymriah coming to market as the first of the CAR T therapies, single-arm launch supported by basically an indirect treatment comparison driven by a meta-analysis type methodology. So real-world evidence is used, but sometimes folks in the space, including on the biotech side, don't always think about it in the same way. And from the clinical side, there's tremendous pressure to focus on the pivotal, but this is the other piece that helps make your product story sticky, especially in an area that what makes cell and gene therapy different? It basically is magnitude of effect and duration of effect, and in some cases like rare disease, you're moving into areas where the burden of illness and the natural history just haven't really been well characterized. So there are many uses here, and we'll kind of stepwise here in a moment talk about some of them. This slide is from a study that was published in Value in Health that I did, where I looked at with colleagues, looked at over 100 health technology assessment assessments from multiple markets and looking at advanced therapies here in particular, and you can see a few things. So there's uncertainty regarding efficacy, there's uncertainty regarding duration of therapeutic effect, and the bars indicate HTAs, like percentage of HTAs that showed this uncertainty. Uncertain link between surrogates and outcomes. If you have a scenario like a rare disease that has a surrogate, but then the real hard outcomes, death, major morbidity issues, hospitalizations don't occur until later, then you have a major challenge often linking whatever that surrogate is, it's a biomarker, it's something else, to those hard outcomes. Real world evidence can help make that case and put the pieces together. Lack of comparative data, study quality concerns, and uncertainty around cost effectiveness, just as some of the things that have popped up in the advanced therapy space as criticisms from HTA agencies in particular. So here in just a moment, we're going to start our discussion, and we hope that you guys chime in, ask questions. This is here for everybody. We want it to be an iterative discussion, and looking at these three buckets again: early development, then getting ready to launch, dealing with regulators and HTA agencies, and post-market, where you're looking at issues like characterizing the patient population, defining unmet need, looking at special trial scenarios, fast-track programs, single-arm studies, areas where real-world evidence can often be valuable. And I won't go through all of this one either, but just to sort of set the stage, where is addressing uncertainty in this space the most important, where you have target population is broad versus narrow. How do you pick your patients? How do you characterize, especially if you're going to have to have a matching strategy with the real-world evidence, and you may construct a synthetic control arm for your study. Can you target the right patients with biomarkers? How well do competitor standard of care therapies perform? What's your differentiation window? And for advanced therapies, you can't just think about marginal differentiation. You have to think about defining what is transformative, and your real-world evidence analysis can help in that process as well. Again, lots of single-arm studies in this space. Generally, everything that's out so far has been launched with early data under that scenario. Will regulatory design address payer and HTA questions? Sometimes your package may be fine for the FDA or for EMA, but it just may not answer the questions that payers have. Real-world evidence can help there, depending on the market. And then what long-term outcomes are most impactful? In this space, you are going to have to design a registry. It's part of what comes with advanced therapies. In this case, and it was mentioned at the IQVIA workshop yesterday, the registries in this space are basically efficacy and differentiation engines that just happen to have some safety requirements, because as you go through your launch sequence and as you push out, the longer you can make the case for how the duration of effect works in this space, the stronger your value proposition is. So you have to begin thinking early about what does your registry look like and how it ties into your overall package. I'll come back to this, but we'll jump right into the questions here and kind of get started. So, given the pressure for payers to limit access to the trial population in the study, figuring out who is in and who is out is absolutely even more critical in this space than it is elsewhere. Would like to just go down the line with the manufacturers here and just take some thoughts about how do you begin thinking early at the design stage, thinking about ultimate market access and commercialization. How do you begin thinking about how and in what ways to use real-world evidence to fill in the blanks? Ramesh, do you want to kick us off?
Well, thank you, Eric. Last 2 to 3 weeks, I've been only thinking about ALS, amyotrophic lateral sclerosis. I can only retain one thing at a time. So I'll talk about my experience on that in terms of early development. We have a program, we are starting a program trying to design the clinical trial for SOD1 mutation form of ALS. ALS is a very, very difficult disease. If you notice that since 94 or 95, the first product was approved, Riluzole, and the next product was approved only in 2017, Edavarone, in this country. And between 1994 and as of today, If you go into clinicaltrials.gov, if you put ALS, you will get close to 520 clinical trials. Everything under the sun has been tried for ALS, and they unfortunately failed. It's a disease that's very heterogeneous. We do not understand the biology of it. So the challenge for us is how to design the clinical trial that can show treatment effect. But more than that, how can we address the payer needs downstream? So the heterogeneity that is present in this disease state is the one that is predominantly leading to these failures. And so looking at the real world data, there's quite a lot of data that is available. There's something called ProACT. It's a database of all the clinical trials where the manufacturers have contributed to it. Of course, it may not fall under the traditional definition of real world evidence because it is from clinical trials, but this is an aggregate data set that is available for us. I recently came across a paper which was published last year in Frontiers in Neuroinformatics, which looks at data from Emory ALS Clinic, 801 patients, close to 4,000 visits, and they considered 38 clinical variables to see exactly how survival is affected by these factors. That for me is a very good example of how real world evidence is used in order to inform clinical trial design. And there's a lot of advances that has happened in machine learning. Of course, they use that. So they use a generalized linear model and a random forest plot model in order to fit this data, complex data, and come up with insights. So that is very important, and hopefully that will help us to design a better clinical trial accounting for the heterogeneity. I always think about the payers, the HTAs. At the end of the day, they are going to ask us to do a network meta-analysis or an indirect treatment comparison. How do I do it? Once the trial is completed, and let us say it's successful, what is that I need in order to do the indirect treatment comparison? I start asking that right from the beginning. The second question is that most of the HTAs will do a subgroup analysis. So the question is, if you don't control for the heterogeneity, the confounding factors can artificially lead to different subgroups. So understanding what has been done using real world evidence is very important to design the proper clinical trial. So for us, these kind of sources that are available in the published literature and the databases that are available will be very, very helpful in order to address the real world evidence requirements, to clinical trial requirements, to design the better clinical trial. It also goes to looking at the primary endpoint. In ALS, almost all the clinical trials use ALS functional rating scale, the revised version. And unfortunately, it is not a very good measure. It does not predict survival really well. So the question becomes like, what other endpoints can we have? Can we have a composite endpoint, including the functional rating scale and the survival one together? So looking at all these different clinical trials and everything that has been published is very, very valuable to design the clinical trial. And last but not least, I'm a modeling guy. So I need to see how am I going to model the states. So the question for me is, will the clinical trial give me enough data, sufficient data, in order to model this disease state? Which means that if I'm going to collect the ALS FRS revised scale, how frequent do I need to collect that? Currently, we are looking at a much greater interval for collection, but I've noticed some clinical trials where the rating scale has been validated even for telephone collection. So we want to collect it frequently because it will help me to design the transition from one state to the other, because it's a nonlinear progression on this scale. So the end result is that all this real world evidence that is out there in the public domain can be really used to refine the trial design with the sole focus of addressing what the HTAs need. The regulators most often, to the extent that you show a statistical difference, let us say in the ALS FRS score, you will get approval. The next question is, so what, when it comes to the HTAs, because they take a much more of a deeper dive. And what we noticed for Zolgensma in Europe and Japan and elsewhere has given us very good insights about exactly how they think, what are the kind of things they look for. So we bring those experience also for the trial design here. So it's never too early to start looking at all these things. And the last one I want to mention before I close on this topic is that In ALS, the preclinical data, the animal data, is not very good at addressing the phenotypes. It's very good at addressing the genotype. So it can tell you whether the drug reaches the target, what should be the starting dose, how should the dose escalation work, and things like that. So it can give you very good safety-related information, but will not give you functional and other information. So this real-world evidence becomes very, very important to supplant the preclinical data to do an effective trial design.
Bosch, do you have thoughts?
Yeah, I'll focus on two areas that we are conducting phase 3 trials in, or will be getting ready to jumpstart our phase 3 trials. And one is in hemophilia, Hem B, the other one's DMD. And in both instances, as me and my team got involved with the early research team, it was so important to see that you need to have a complementary real-world evidence program, whether it is just, again, basic study design, endpoints, type of patients, to actually developing a registry or conducting a study in parallel to look at the impact on standard of care, and to, why should payers pay for these expensive medicines? So case in point, hemophilia, this is a disease where there are many alternatives currently in the marketplace. There are short-acting agents, there are long-acting agents, and while the larger payers, they actually wouldn't mind having a gene therapy on the market because it's cost savings. It's highly cost effective. But when you look at smaller employer groups, you look at mid-sized payers, you look at particularly Medicaid, there's going to be a huge budget impact when you bring a curative agent into the market, whichever company brings in first, because it's going to affect their budgetary cycles in a strong way. So a couple of areas, as we devise payment options, what's going to be affordable, we also have to look at PMPM, PMPY, whatever the payers use to look at budget impact. And that means you've got to look at the total cost of care that they're paying for this particular patient population. The second thing is what outcomes are important. And it's not just bleeding rates, it's also what a patient goes through when they have hemophilia, their inability to travel, their inability to play sports. And all of those, which we call patient-centric measures, they have to be collected, and I'm just using the terminology real-world evidence in a broader fashion to say you've got to collect that data from patients through either natural history studies, through patient interviews, and when we just think of designing a registry to look at real-world outcomes, the patient-centric endpoints are also an important component in the overall picture. So we can, when we go to whether it's ICER or whether we even go to a commercial payer, we have to kind of show the impact not just with economics but also with patients. It becomes more challenging when you go to a disease area like DMD, where there isn't really a strong standard of care. These patients, it's a chronic fatal disease. Adding a gene therapy is going to be fantastic for these patients when it becomes available. But the payers are a little concerned because the time frame when Pfizer and other companies will bring DMD to the marketplace, there will be several other gene therapies. So you've got to kind of look at just the overall, the pipeline of gene therapy assets that are coming in and how they are going to impact a payer's budget. And so the two aspects of conversations we continuously have with payers, both in the US and Europe, is what's in the pipeline for gene therapy, what are they currently paying, how is Pfizer going to partner with them for risk sharing, for outcomes-based guarantees, and those need to be thought of prior to a phase 3 or in parallel to phase 3, because if we don't have that, for a company like Pfizer, where we have 5 business units, we've got oncology, I&I, our rare disease, all competing for a central pool of funds, we've got to be able to explain to our management, to our board, why an investment in gene therapy makes sense. And that includes the payer piece of it, and we gather all the insights from the payers' real-world evidence data sources to build our pricing and value proposition strategy. And then we make sure that the real-world evidence plans that we are put into the operating plans, 5 years prior, they actually get executed. It's very complex. It takes time. They're expensive. But if we don't do it, then, Eric, you had some slides on some early gene therapy assets. None of them yet have been a blockbuster. And we just need to make sure that we are preparing the marketplace, our customers, particularly our payers, just to understand that an investment in gene therapy is going to help them manage their budgets. It's going to help take care of the patients. It's going to be ultimately cost effective and sometimes cost savings.
And to the point about payer awareness or stakeholder HTA awareness, even in this space, some of the HTAs are becoming more aware, but with ARM and National Association of Managed Care Physicians, we've had a couple of collaborations that have resulted in papers. One was published last week that's a reimbursement roadmap, but generally, when we've presented the ARM statistics on the marketplace, how many clinical trials, how many therapies are close to market, and those types of bits of information, largely the U.S. commercial payers until recently were unaware. They have no idea what's kind of coming. So it's been really interesting to engage with them and really talk through what's different, and then how many of these might you likely be seeing, because here in the next 3 years we have the potential to triple the number of therapies that hit the marketplace. And Francesca, I'll ask you, because in your pipeline in general, there are different things, but one of the things that is a little bit different from a hemophilia is you have diseases that have long-term episodes before you really start to see hard outcomes. And in some cases, there are things that are hard to measure. How do you begin thinking early about how to define a transformative value proposition when you have diseases that may not unfold? It's not like SMA, where the patients really may fade fast in some cases. When you have these longer-term rare disease scenarios just in general, and you also have patient-centric measures that may be involved, how do you begin to think about that from a real-world evidence perspective, just in general? Because
Right.
It's a different animal, it's a different kind of thing to solve for, I think.
Sure. For example, I noted earlier that we have both pediatric rare diseases as well as large retinal diseases. On the pediatric side, MPS II, for example, we're developing a therapy that would really be focusing on the CNS effects of the disease. There's already an enzyme replacement therapy available now that's used for more of the somatic. And so when you're thinking about how to affect change sort of on the neurocognitive level, then that's something that's likely to take a long time to demonstrate. And so we've done sort of a number of things, at least in our early thinking and our early exploration, in trying to identify not only will you have your primary and secondary endpoints that will be looking at neurocognition, but how do we think about demonstrating value and effect that can show the meaningfulness of these changes that can take a long time to manifest. And there are a couple of things that we've done on the MPS side and that we're trying to get started very early in development. So we're in phase 1-2 for MPS. And so it's a disease where there is not natural history available. There's not a lot of data on burden of illness and resource use, at least in the US. We saw that there was some literature from Europe, and so we took a study that had been done in Europe looking at sort of burden of illness in terms of resource use, education, impact on employment, all of things that are very impactful in this disease. And we adapted it for the US and did an online survey just to get a sense of the burden there. And we worked with a patient advocacy group and also with Invitae, which has a registry of patients in MPS. And we fielded the survey both in the US and Canada, and we had an enormous sort of response from the community in terms of, we had to shut it down within about 72 hours, which was far quicker than we had anticipated. And as one would expect, the burden was very high. But what we also learned from that when we did our abstracts and posters, my colleague who was presenting the posters, there was a lot of patient and family expressions of thank you for getting this information out there. And so what we're learning is that there's more that's happening in this community that they want to see impacted and that their voice isn't always being heard. And we also did a number of interviews with caregivers of these children. And while we had always planned to include very early on sort of traditional validated quality of life tools in our clinical trial, what we also learned by talking to the families is that some of the things that were most important to them, things like toileting or making sure that the morning routine, brushing the teeth, getting out of bed, things like that, was very important in that their child could sort of initiate contact or cuddles or conversation, that the things that were really important to them aren't always included in those traditional validated tools. And so those are the types of things we want to ensure that we collect over the long term. So thinking more creatively and innovatively, video and other types of things that you might want to incorporate early on that you can learn from, and then make sure when you think about your PRO and other tools going into your pivotal trials that you've identified those things that are most important and that you've got the right tools that you can incorporate to gather that data, but you're starting that in your phase 1/2, so you have the longest amount of data possible to have that at the time of launch.
And that raises a couple of points, I guess. With payers, when you use different instruments, especially if they're unfamiliar, if there's something where it may not have been built for a particular disease area, part of the challenge is that maybe in Europe it's better, but in the United States, if you have something that reads out at a 0.75 versus a 0.78, a lot of the payers don't understand what these instruments mean, and you have to think, how do we begin to map that or collect information that makes it real? What does this mean the patient can do? Can they walk up and down the stairs? Can they walk on a treadmill for 6 minutes? What are they actually able to do, and how does it make it real for their life experiences? And the other bit, which comes along with anything in this space, is that with the price points we see in the marketplace for advanced therapies, as well as the whole issue of needing to demonstrate both magnitude and duration of effect, you're looking at a comprehensive story. You can't take anything for granted in this space, I don't think, because your value proposition may turn on a number of little things. It may not turn on any one thing. And so with all the examples so far, you hear how the manufacturers are thinking, or very early in some cases, about how do we tell this story, kind of going line item by line item by line item, and looking at what is differentiating. It's different if there's nothing available versus if you have a fairly crowded active space like hemophilia, where there are fairly successful therapies. They're not perfect, but they're available, versus an area like MPS or originally SMA. There are two therapies now, but it's a different sort of scenario of how you think about going through your value proposition development. One of the questions that I would like to pose, even for so-called monogenic diseases, and I come from pediatric medical genetics background, when you open the hood and you look under a monogenic disease, what you often find is that there's a lot more going on than you think that may impact patient response. But many diseases, oncology, very polygenic, and in some cases we're looking at solving a shotgun genetic problem with the rifle with how we develop therapies. How do you guys think about early, do we have a biomarker issue? Do we need to think about a backup plan? Because what happens, and we don't know with some of the cell and gene therapies, if there's a differential response where you may have a super responder versus a normo or not so attractive responder. Are you thinking about dealing with the biomarker issues along the way? Any thoughts there? Any of you? Whichever feels like jumping in.
I can go back to my DMD example because we're in
sure.
the thick of it. The biomarker is minidystrophin, and even in conversations with regulators, leave alone payers, I think the feedback has been that this has to be more outcomes-oriented, not just the biomarker. So there are generic outcomes, like or questionnaires or evaluations like 6-minute walk test. But when you're looking at, say, a 5-6-year-old young child, we realize that a 6-minute walk test may not be adequate. So we're looking at other outcomes, more patient-centric, that go into more specifics around neurocognitive functioning. And then the challenge to that is also you have the ambulatory patient population, non-ambulatory patient population. I think the younger you treat these patients, it's easier to show effect size and improvements. When they're in a non-ambulatory setting and they're wheelchair bound, then you have to approach it differently. And that's when, as we build that whole value proposition, the evidence, we've got to kind of, we put on the payer hat to say why would they, what sort of pushback we could come, because again, this is not going to be looking at the Vexus model for Zolgensma. You can look at certain endpoints or outcomes like sitting up or being able to ventilate is sort of the last step, but sitting up was one of them, and that's the sort of health states we are considering. But all of that has to sort of come in as we build the economic models from either existing data or literature or prior trials done in the DMD space, our own trials with domacrozumab, which didn't make it, as well as actual natural history type studies or getting the data from patients and families alone. And that takes a while. I mean, I would say this takes as long as a clinical trial does. If again, you want, if this is, the price points are in the chronic illness kind of state, then I think it would be okay to be having a less rigorous approach. But when you're looking at gene therapies, high price points, when you don't have a direct cost offset story, you've got to look at the totality of the picture and across different target patient populations to explain to payers and to HCPs why they would give a gene therapy especially to the non-ambulatory patient population. So especially in those areas, I think biomarkers have benefit, but negligible benefit in the eyes of the payers.
Sure.
And even 6-minute walk tests, 4-stair climb tests are very, very generic. You've got to get much more specific.
And I would just add to that, because I think this sort of illustrates sort of a critical issue between what you're designing for the trials and what payers will actually look toward and use for measurements. And I know we're going to talk about post-marketing, but I think it's important to say here that the payers looking for things that can be measured and will be measured by providers primarily. What a biomarker would make it simpler, but in reality, this doesn't exist for a lot of things. So I think there's really a challenge here to start to bridge, to think about what is the equivalent of the 6-minute walk test, as an example, that a provider will do in their office reliably, that can be captured and reported back. Providers don't do 6-minute walk tests outside of a clinical trial. So that really is a challenge. And when you move that next step from provider-collected data to patient-reported outcomes, payers have an even harder time with credibility there. But there's a whole science around validation of reporting instruments that we really need to get on top of. And one of the things that sort of strikes me is even in these chronic diseases that are very different, there could be a common patient-reported outcome tool that could work across many. And it really would be beneficial to all to improve the quality of data that's collected in the real-world setting, so that it can be more useful down the road by payers and others.
And we're jumping ahead here, Jane, but MIT, you may want to speak a bit about the kinds of things you guys have been doing. And when I say jumping ahead, I know you guys have done a good bit of work thinking about risk-sharing agreements, and the point that may be important is in early development, you have to start thinking about what may ultimately your risk-sharing agreement be based on, what it turns on. Is it a biomarker? Is it something you can measure? Any thoughts there? And just maybe with the preface of what you guys have been doing in case some folks don't know.
Sure. So as I said earlier in my introduction, I'm a senior advisor to the MIT New Digs Focus Project, which is really around how are we going to make sure there's sustainable access to these therapies for the long haul, that payers are going to cover them, the reimbursement will be adequate, so we can keep fueling innovation to help patients ultimately. So the work has been done in a multi-stakeholder fashion. So that includes the payers, the developers, the patient representatives, policy folks. Everyone that is, touches this has come into play, including reinsurers and others. So the thinking is, again, with that broad slate, is how do you get at ensuring there's reimbursement? We talked yesterday, so a lot of the different discussions touched on some of the risks that to reimbursement, like the payment timing being one time, very high costs relative to our current model. The actuarial risk of, am I actually going to have one of these or 2 or 5 patients in my plan? And what is that going to do to me from a budgetary standpoint? And then the uncertainty around the durability. So all of that has really led payers and the effort that's gone on at MIT to think more and more about performance-based contracting and different kinds of precision financing tools to help ensure reimbursement that's sustainable. All of those performance-based options rely on metrics. And as we've been discussing down the road here, these metrics are challenging. And what we found in some of the early piloting work is it's difficult to translate that clinical trial to real world. And over time, how you measure that performance. I think, as I alluded to, we're missing some critical tools in validated patient reporting outcomes data, testing that would be appropriate for a provider to do in their busy clinics on a real world basis versus a study basis, and so on. But those are really important factors for not just performance-based outcomes, but also in some cases for just the coverage to begin with.
And with the first wave of therapies that launched onto the market, their risk-sharing agreements weren't terribly complicated, frankly. I mean, they were very simple. Some involved a payback if it didn't work, but it was usually a short time to demonstrate that it hinged on. We're getting ready to begin testing the more amortized, the multi 3 to 5 year payback models. Any initial feedback from your MIT payer groups on that? The short-term payback approaches have seemed to be reasonably successful, but any thoughts about challenges that they've raised and how it may help think about the metric that you would use?
Well, the biggest challenge to the multi-year type of agreements that go out 3 to 5 years is the ability to track the patient. And that brings up a lot of different issues. If you're a patient who luckily is truly cured, why do you want to keep going back to the doctor as an example? I mean, there's just some basic things in this. So is the doctor, is the patient going to be amenable to follow-up? Is the doctor going to, want to see a patient that looks, that has been, cured? What's the doctor going to test? Where does all this data go? So we have work going on. I won't say it's completed yet, but it's really in the works. That's looking at what's really required. And I think about it in sort of 2 buckets. One is the what. What's the actionable evidence? And then how do you, what is it and how do you capture it and how is it available? And then the other is the how. So that's the efficiency of being able to capture it and your effectiveness of being able to capture it over time. And then the other piece that I would add into it is the scale. And I just personally don't think that it's going to be a very good use of our healthcare dollars to have every manufacturer create a registry and a database and replicate that across all of these different diseases. But are there ways that we can think about scaling this and across common diseases or even across, all of these therapies. How do we think about that in a more meaningful way? And then the third area is the policy piece of this. So does our, HIPAA is 20 years older now or more now, and it really didn't envision the kind of things we're talking about today. And how, what needs to happen from that standpoint so the appropriate data can be used on an ongoing basis. So I think I just opened a can of worms.
That's okay.
Yeah.
Can of worms is welcome. It's perfectly fine.
Yeah, I think I would just add to that I think that as companies, we're also thinking about that very early in development because we are capturing data from the very first patient in on that very first phase 1 trial. And you'd be collecting that not just sort of a year out from the, as your trial ends, but you'd be collecting that all the way through for 5, 10, more years. And so we're collecting data, not just, the traditional safety data, but there are other things to think about in terms of, longer term quality of life or sort of burden of illness, resource use, things that you want to start collecting sort of early to determine the impact of your intervention. And you already don't want to burden these patients for years on end with, long sort of questionnaires or touches back to the healthcare system. So we're already starting to think about how can we sort of minimize that impact on the patient and their families early so that they're willing to continue to participate, and then that just continues as you're thinking about sort of the long-term sort of post-launch and demonstrating your durability.
Okay. Any other thoughts or?
Going back to this durability issue, actually, if you look at the ICER SMA review, the base case for Zolgensma versus best supportive care, the incremental cost effectiveness ratio is $243,000 per quality adjusted life year. This is assuming the horizon is lifetime. That's what ICER provides for ultra-rare disorders within their framework. And the durability of effect for Zolgensma is lifetime. Now, they ran a scenario analysis, and in the scenario analysis, they limited the horizon to 10 years. So effectively, the duration of effect is 10 years for Zolgensma. The $243,000 per quali actually more than doubled to $525,000 per quali. The reason is, if you look at the quality adjusted life year, it dropped from 12.23 to less than 5, almost a 60% drop, because a lot of benefit happens after 10 years. The patients sit, they live longer, the median survival is 30 years, and patients walk. 16.67% of the patients within the data that we used, ICER used, they walk, they have a normal survival pattern, and they have greater quality of life. So that is why the quality of life drops tremendously. However, the total cost, 80% of the total cost incurs in the first 10 years. Only the remaining 20%, because it's a supportive care cost, because of patients living in the sitting state, the incur cost happens. So the costs are upfront, the benefits are over the long run. So durability of effect becomes very, very important, as this example shows. So payers have a very good reason to ask the durability or demonstrate the durability of effect. So what we are doing is right from the beginning, as parallel to the clinical development program, we started thinking about registry. And to be really useful, we have come up with a plan. It is already implemented. We call it the Restore Registry. It is a disease registry, and it's global in nature. It is prospective, observational, minimum intervention in the sense that patients don't have to do anything. They have to enroll in the registry and be under the usual care. And to address Jane's thing that if every manufacturer were to have the registry, it will increase the burden of data collection for the physicians, patients. So we co-opt. So that is any patient that goes into our managed access program or a compassionate use program, we let them come into the registry. Also, we work with other established registries like Cure SMA, ISMAC, Treat NMD, all this data to flow into our registry. But the bottom line is this registry will help demonstrate the durability of effect. And it is important in the US, but even more important in Europe. One can say that IFA has their own registry and they use it for managed entry agreements. The problem is, just last week or 2 weeks ago, I reviewed a paper on managed entry agreements in Europe, and they noted that none of the results from IFA's registry program have been published. So we really do not know how effective these registries that IFA has collected over a long period of time has been in what you call monitoring the disease progression, how effective the drugs have been, so on and so forth. So there are some limitations even in Europe with the registry collection. So hopefully these kind of restore registry that we have, which we started very early on, will help address those kind of questions. So it's very important for me, essentially it's a duration of effect. And the second thing is that the registry will collect all the relevant information. It will have the patient baseline information, but it will also collect information on nutritional, respiratory, milestone achievement, quality of life using CHOP indent and other measures. It will collect the caregiver burden. So all these things are basically built into the registry so that it is not just looking at survival, but also other functional measures to the extent that you can use that to contract upon for your performance-based agreements, especially in Europe.
The ICER report for Zolgensma was an important milestone. Any sorts of insights into, aside from the pivotal data, how real-world evidence was helpful in understanding the whole picture? Anything, the takeaways there?
I can tell you, July of last year, end of July, ICER came, called us and said, we are going to do ICER SMA review. So the next 9 months was a marathon at the rate of a sprint run. I think it was very fundamental because if you actually look at the disease progression, we were looking at type 1 symptomatic SMA. And the natural history is patients are in the not sitting state. They are born in that state. And a first respiratory infection, unfortunately, might kill them. They die. Or they survive that, and then over a period of time, they get into permanent assisted ventilation. And then they die. So the natural history is not sitting, permanent assisted ventilation, and death. These are the three. But under our treatment, and also under the nusinersen treatment, a proportion of patients essentially go to the sitting state. And in our case, 2 patients out of 12 ended up walking. Now, no amount of natural history data is going to give information for the sitting patients and the walking patients. So where do we go? This was a dilemma. So we spoke to a lot of internal as well as external clinical experts, and then decided that we will use type 2 patients whose maximum milestone they achieve is sitting, and type 3 patients whose maximum milestone they achieve is walking. We will use them as a proxy. So the natural history for type 2 for sitting patients under treatment, and the natural history of type 3 patients as a proxy for walking patients, we will use it. So we went back to old registry data that was published, Zeres paper, from using Polish and German type 2 patients. We used it for the walking, sitting patients, and similarly existing registry data for type 3 patients. We gave this information to ICER. We told them our dilemma, and they thought it also kind of a very most sensible thing or most conservative thing to do. So they ended up adopting that assumption. So without registry, we could not have done the modeling or showing the disease progression. Of course, one can ask the question, can the patients do better than the non-treated type 2 patient, the sitting patients on treatment? Or can they do better, the walking patient, can they do better than the typical type 3 patient? We do not know. So ICER ran some scenarios looking at they actually ran only the pessimistic case where they drop milestones and things like that, but the registry data will provide more information down the line, whether our assumption was a very conservative one or, or is it very optimistic. That will provide that answer. But without the registry, we could not have done that.
So with just looking at the United States, just shifting gears just a bit, with the 21st Century Cures Act, where it's opening the door for use of real-world evidence at the regulatory level, what we're starting to see is evidence development is now certainly a journey. It's not a destination. It's not like we plant the flag and we're done, reach way back, pat ourselves on the back, our work is done here. It's a continual evolutionary process now. with the 21st Century Cures Act, not to get into the details of it, but and Bosch, I'll direct this to you. How does a large organization like Pfizer think about, hey, now there is an opportunity to provide this evidence and the world is evolving. How does a large organization think about the opportunity versus risk issues with the uncertainty of what kind of evidence is acceptable? Any insights there?
So a couple of things around that is, and I'll use not a gene therapy asset, but something that's currently in the marketplace called tafamidis or Vyndaqel that was launched by Pfizer earlier this year for TTR cardiomyopathy. TTRCM is a form of cardiomyopathy that's caused by amyloid deposits. And usually amyloidosis could occur anywhere. They get deposited in the liver, in the tissue, but when they get deposited in the heart and lung, and the hearts particularly, it leads to cardiomyopathy types of symptoms, and ultimately these patients end up, they're older patients, Medicare primarily in the United States, and they end up having premature death and disability. So our trial was in TTRCM patients who were in earlier New York Heart Association class 1, 2, 3, but where I think the 21st Century Cures Act could help is really to understand the value of these kinds of treatment early on in the disease time frame. Our current label indication is really for once they're detected, once they're diagnosed, they are in NEHA 1, and that means they're fairly advanced disease. And then when you start treating them, they have a limited lifespan. You're extending life, and going by the principles of cost effectiveness, these are older patients. The survival, it's sort of like oncology, even if it is, a couple of years, it still isn't enough for these treatments to be cost effective because you can't really show a 10-year survival benefit. These patients were mostly in the 70s, in our trial, the late 70s. What we have been able to show is a 30% reduction in mortality, which is great given that these patients are older, given that these patients die at a faster rate than patients even who have bladder cancer or prostate cancer. So I think we're understanding with the partnership of COEs that treat TTRCM, if there's a diagnostic pathway to find risk factors that are associated with TTRCM, and following again the natural history of these diseases, when could they benefit from treatment is an opportunity for Pfizer to use real-world evidence. We are not there. We have to kind of follow what the labeled indication is. But I think just again, given that that many patients are just misdiagnosed or cannot get identified early, is unfortunate for the patient. So that's something that we would explore with the FDA to see if we could study that through real-world evidence study.
Thank you. I'm going to, yes, question in the audience. And we have a very strange cue that we'll need to talk to here in just a moment because they're recording the sessions. Right here.
So if you're talking about real-world evidence in TTRC and trying to figure out how to get evidence for younger patients to be able to intervene earlier, physicians are going to use your drug for older and younger patients. They may not, payers may not be anxious for them to use it for younger patients, but they are in fact going to prescribe it, and some of those patients are going to get reimbursed. Do you have a method now for capturing data on those earlier patients and using that as real-world evidence? For example, could you, as part of a contracting process, say, look, we'll give you some kind of risk share, but in return, we would like to see the data not just from those patients who fall into the label, but also for those patients who may in fact get the drug and may not be in the label, but whose data would be valuable to us and indeed to the world in general.
Yeah, so we do have a hub because of the complexity of the out-of-pocket costs for these patients. They're primarily all 65 plus. So there is Medicare, standard Medicare. There is LIS. It's a whole host of different, payer channels. And we do have a hub through which we actually know, we can track, again, where the patients are coming from, a little bit about, the comorbidities, because a lot of them are going through utilization management. So we are in conversations with payers and also with some of our KOLs and thought leaders around, because we want to, again, make sure that there is a diagnostic pathway through scintigraphy, as you may know, that these patients could get detected early. And so following AI logic, and that's again real-world evidence around which patients should be considered for scintigraphy, that's something we are beginning to kind of educate heart failure specialists on. And through that, we are able to determine, again, what % of the patients are going to be earlier, younger patients. And we are beginning to have conversations, but not just with payers, but also with some of the academic centers of excellence in making sure that these patients get detected early. Because once you're in New York Heart Association 2 or 3, it just gets, it's an irreversible disease. This is not a gene therapy. So what Vyndaqel can do is prevent the progression of the disease. It cannot reverse the disease. So we've just begun, since the product was just launched a few months ago, we've begun to have those conversations on identifying those patients earlier.
And with some of the oncology examples in the slides starting, there's an increasing openness to look at real-world evidence in terms of supporting label expansion steps. We're seeing more and more and more of it because I think stakeholders are beginning to recognize that there's good enough evidence to inform certain types of decisions. It's not a crystal clear black and white scenario, but there are plenty of examples of using, what would then be sort of off-label data just because of the way prescribing happens and other stuff to begin to fill in the blanks for some of that label expansion activity. Jane, did any of that sort of thing come up in terms of label versus off-label use, or in terms of missing evidence gaps that could have a real-world evidence solution in some of the focus work that you guys have done? Was that something like a crosstalk between payers and providers, or any insights there?
Well, probably more in the opposite direction, which is not what people want to hear. So again, that work is really ongoing, and we actually have a meeting in a couple weeks where I think we'll get deeper into that. But I think in general, the payers, as a whole, while they're afraid of the financial ramifications of this and trying to figure all that out, they actually are very excited about treatments coming that have real benefit. And we did a survey of payers that covered 280 million lives recently. And the payers want to pay for what works. And so if there's credible data that can show that the treatment works in a population that's broader than the label, I think payers would be open to that. But what they're really looking for is not just like a conjecture or the provider saying, I don't really have any other choices. What else can I do, right? So I want to try this. Not really the trial and error, but systematic collection of data that helps to support a case for extension of the label use, is, I think payers not every payer, but many, many payers will be open to that because they'll see the value of that in their populations.
And running slightly counter to some of that, I think when you talk to payers early in this space on the front end, what you kind of hear and what you see in some of the coverage policies, at least in the United States, is that there's not a whole lot of faith-based reimbursement decisions, that whoever's in your data collection, that's it, right? So they're not reaching outside. If you look at Spinraza initially, at launch. Some of the payers looked at the data, and even though the label was a bit broader, their coverage was a bit narrower initially. Now, some of that has been filled in over time, but at the time of the initial coverage decision, they'll make narrower coverage decisions than what your label is if they don't believe there are enough patients in a certain bucket or you're covering certain things to their liking. One of the things that with the recent ARM NAMCP paper that we found is when we surveyed about 45 payers, what we found that give or take, about half of them have not yet, even for products that have been on the market for 2 years, rendered a coverage policy. So part of it is, even though the payers say, yes, we want transformative therapies, we're tired, we don't want the next antihistamine therapy out there, we want something that really changes the game. At the end of the day, it looks like they also are on the same token dragging their feet in making some of these decisions. Now, I don't know whether that's an educational issue or whether that's just a convenient delay of decision making or what it is. But it seems to be the outcome, because I hear them say what you just said, but then you see them do something that may be slightly different than what they're saying. Any other questions from the audience now? Yes. Wait for the boss.
I've got one for the pioneer manufacturers of drugs on the market. I should say I'm a colleague of Francesca. I'm wondering that if, let's say you have a conventionally delivered biologic on the market and a gene therapy version is under development. There's a lot of information about the first generation products in the FDA drug master files, most of which is trade secret. Do you think there's any way that incentives can be given to firms with products already on the market to use some of that data as real-world evidence, historical controls for the gene therapy generation of products? Because right now, both in the generic and biosimilar markets, the competitors, these copiers have a right of reference, but they don't ever get a right to see the data. So I'm just wondering, have you thought about that, or other stakeholders thought about using some of the data that is in the possession of the government, it's the property of the developer, but could be very useful in providing confirmatory data rather than reinventing the wheel with confirmatory trials?
I think it kind of goes a little bit back to, if you look at it just from the registry standpoint alone, the debate between should we have a product registry or should we have a disease registry. With the disease registry, it begins to make more sense, minimizing burden of collecting the information. It may be more cost effective. But on the other side, you have less control over how the data is used. There are some initiatives, and I'm not sure if there's anybody here from Global Genes in the room, but there's an initiative that's a pretty ambitious one that they're helping lead to begin to connect the dots and make some of that data more available. I don't know if there's anyone here that could speak, but basically, so sometimes things are moving in the direction of being able to have broader access to data, and in other cases it's more complicated. I don't know whether any of the panelists have any thoughts there, but it's a kind of a catch-22 situation.
I think it's not yet there. I think it's harder to do it, as of even today, because of trade secrets, as you said. I can say from the Pfizer perspective, we had the readout from a sickle cell disease program recently from Rivapancel, which the study was designed to manage vaso-occlusive crisis in sickle cell patients. Unfortunately, the trial did not read out to be positive, but we got immense data on the patient journey, and this is a hard to even access patients because they are sort of the youth in compromised neighborhoods. They go to the ER often, so there's a stigma associated with them. They often do not go because they just don't want to deal with it, and they manage the pain at home. But what we found through the work that was done several years ago and around standard of care and how these patients are being treated, that's changed substantially to where things are today in the management of sickle cell disease, with more outpatient centers opening up. And that's a very important finding for all companies and manufacturers who want to continue developing assets in the sickle cell area, particularly around VOC. So at least from a Pfizer perspective, we will publish the data. We want to present the data, and more so for our patients, our physicians who are getting engaged in other clinical trials, or what the standard of care is today, and why rivapansel was unsuccessful is because the standard of care has just shifted, and that's a great learning for, I think, every stakeholder to kind of know, because it's expensive to do these trials, and it doesn't help the patients when the drug is unable to read out positive.
And on some level, I think that we're also at the beginning of another journey too, right? So if you look at what's happening just in the data world, we're having multiple companies that have different platforms for rapid data analytics and AI, and we're starting to connect the dots, and we're starting to try to think about how do we get to some common data architecture so we can compare things across. I think that once that really does, and there's a long way to go, but I think once that does pick up a head of steam, some of that issue that you ask about begins to go away, because we'll then have easy, faster access to real-world evidence in ways that we never did before. So one example is, have recently been looking at oncology data sets that have clinicogenomic information that's also married with longitudinal real-world data. So if you're developing an oncology product and you're going to launch with a single-arm study, a tremendous difficulty is being able to find the biomarker tag match patients to be able to make that case. Ultimately, some of those bits will be connected up, and they have very different types of biomarker tests. Is it a foundation one test? Is it a single marker test? There are probably more holes than cheese across some of these data sets, but ultimately we're going to begin to connect things up where that data becomes more accessible more rapidly, and I think that will help solve some of that problem, but there will still be many gap steps along the way, dealing with the current state issue that you raise.
I just think technically feasible with AI, it's the trade secret. It's going to be a barrier, so I think maybe it's time to start thinking about incentives.
Yeah, incentive, sure.
Mm-hmm.
Yes, sir. I have another question over here.
I'm Aaron Stein from Sangamo, and I think as we look at the advances that are being made in gene therapy, currently we're comparing gene therapy for reimbursement compared to existing standard of care. It's not going to be too much longer when we're going to have gene therapy versus gene therapy. So you can't do comparative trials. Well, I don't think you can in rare diseases. So how do we use our RWE to be able to try to demonstrate some additional benefit to your therapy versus what might be out there in the future?
Any of the panelists want to take a crack?
I can talk about for Zolgensma. That's a very good question. At least what we did for FDA submission, there was natural history data, PNCR, so we kind of case matched it, controlling for the SMN2 copy number and the time of diagnosis. So we created a natural history data set that was comparable to the patients in the STRIVE and the CL101 trial, the START trial. So we provided that. But the question of doing network meta-analysis or indirect treatment comparison is a very important one. And that's why right at the beginning, when I talked about the ALS program that we are thinking about, I said that is going to be one of my important issues. The issue is that if you go back to older data, the standard of care is changing. It's evolving. So most of the HTAs will say that that is not relevant. So you need to bring very recent data. So when you want to compare one gene therapy versus the other, it becomes tricky, especially if they are single arm. And ideally, you need individual patient level data to even start comparing, if it is possible. You can control for the other factors, the confounding factors. But many times we don't have it. So on the other hand, if it's not a single arm data, if you have a control arm, then even then it becomes difficult because your control arm and the other control arm, the placebo arm, may not really match. And IQWiG has shown that when I think BMS submitted the data for Yervoy for metastatic melanoma, they did an indirect treatment comparison with Zelboraf, and essentially IQWiG heaped a lot of criticism on that because the baseline characteristics for the Zelboraf's placebo arm and that of BMS did not properly match. So it's a very tricky thing. It's very hard to do. But definitely the questions do come up. So I think it's better to start thinking early how to do it. For ALS, it's slightly different because you do not have that many approved therapies or effective therapies. So it's a slightly different issue. So every disease state is going to be different, but important to start talking early because IQWiG and other, even France for that matter, they do request indirect treatment comparisons. And when you have gene therapy with almost similar mechanism of action and going after the same target population, it almost is a given that you have to do it.
And
Yeah,
go ahead.
I would just add, like, even looking, and as a neutral third party, looking at the SMA report, where ICER looked at Spinraza, and then they looked at Zolgensma, and the conclusions were very different. And so I think, especially in the heme space, there are going to be, it's not going to be one product, there'll be at least 3 that I'm aware of, maybe more. And it's going to be just a natural, even with all the technical issues that you just talked about, I think payers are just going to compare one versus the other. And there's going to be subtle differences, to be honest. And if we, we have to have, again, it's probably going to, price is going to play out ultimately. And then, in terms of efficacy, which one's better. But again, so the indirect comparison has to be very carefully done, because if you just start looking at ABRs and bleed rates, and they're just subtle differences, then the payer is just going to go down that whole tunnel of, give me the highest possible discount.
Right. I think it may be this is an area where patient advocacy groups and others can come together. I think in the hemophilia area, there is an external group that has worked with a number of different manufacturers to identify kind of the key areas on which to evaluate the therapies. And so you do have sort of at least a little bit of bringing together around some consistent metrics, and perhaps in areas where we know that there'll be multiple competitors coming to the marketplace, then there's an opportunity for an external third party to sort of bring those manufacturers together in a broader community to think about those areas that are most critical to measure that can be compared across.
And I think my answer to your question would be it depends, right? I think that from the clinical trial standpoint, there are enough dissimilarities of design that it makes it very difficult, and some of that may be by design, to compare things. Some groups will focus more on certain types of outcomes as part of the differentiation strategy than others. You have to drill down to almost the chart level, which gets to Ramesh's point about going to the patient level to pull out outcomes data. You then first have to, in rare disease, have to be able to find the patient, and those patients may be strewn across a number of different, you often can't get it from data sets. Some of the data sets may not be broad enough to have enough of a sample to really match up and look at what's going on. You almost have to do, go to the chart level to be able to have a first chance at pulling out that kind of level of specificity to line the story up and really have your ability to make that real world evidence case. Maybe at the registry level, if you're lucky enough, you can have that information, but it is complicated. All of the indirect treatment comparisons that are meta-analytical, there have been evolution of other methods that take into account some of those differences that try to address the variability, and they've been increasingly accepted by HTA agencies in Europe, but it's still it's not for the faint of heart. It is still complicated. In some areas like oncology, where the outcomes you're measuring are more similar, it may be a little bit easier on some level, with the exception of matching to the right patient groups and other stuff. But in areas like rare disease, it is very difficult to do those kinds of comparisons just because of availability, sheer availability of laying hands on data. Any last question, and I'll give the 5 minutes. Any last question from the audience? And then we'll have one last say of the most important lesson that you guys think you should impart to the audience. But go ahead, sir.
So in terms of deciding what's in your evidence package, how do you involve things what the actual patients and their families care about? And I'm thinking things like the ability for the patient to prepare their own meals or brush their own teeth. And how do you convince payers that that's actually important?
Yeah, that's a good one. So I think I talked about an earlier example where we had done some work with families where we'd identified some of the areas that were very important to families, which were a little different and weren't captured adequately in some of the traditional validated quality of life tools. And so I think at least we've tried to sort of try and identify some of those areas early, so and explore them early, so that depending upon early data, we might be able to actually consider sort of a formal validation process and working with FDA to see if you can actually, validate a tool that then you would then employ in your pivotal trials. So it's a long and arduous process and not always clear, but we certainly are very committed to ensuring that what's important to patients is incorporated into our trial in some way. And I also talked about, there are lots of there are wearables now, there are videos, there are other ways to capture data as well that can provide a more full picture of the kind of the meaningfulness of the intervention and impact on families, which I do think is important for payers, and perhaps it's just supportive in terms of your FDA or regulatory submission, but you can publish those data. You don't need that same, perhaps, level of validation to take some of that information to payers if you've collected it over time.
Yeah, and I'll add that when we start designing our clinical studies, as early as even phase 1, we do bring patient and family input into the design, into the protocol, into the endpoints, and then there are existing measures that can probably capture, but especially in hemophilia, there are lots of questionnaires. But when we find that there's a gap, we've actually developed exploratory endpoint questionnaires that would have to get validated through the trials because we just don't have time sometimes in rare disease to kind of do an actual validation, put it out there. I think the totality of the evidence is very important in payer discussions, whether they make it to the label or not, because it captures things like what you said, making breakfast, being able to kind of, in hemophilia, play sports. And you were talking about gene therapy, but I think what our plan is also then to make sure as that questionnaire becomes more robust and we begin to get data, could we put it in a registry and could we collect that just, once a year, once every 6 months? You don't have to kind of collect it all the time because hopefully these patients are cured and normal, but you need to kind of keep building that sort of, the evidence around the longer term impact, the durability, outside of just bleed rates and outside of factor levels into what this means for patients. So we began to think that, execute on that. I have to tell you this, as when we took tafamidis, which is not a gene therapy, but it's a drug for ATTR-CM, and we had 6-minute walk tests. We had a quality of life instrument in the trial. Both of them are in the label, but the payers just don't care about 6-minute walk tests. And of course, this is an elderly patient population. Their response was, okay, you want me to pay for this drug so that grandpa can walk X number of steps. So you've got to quantify that in terms of, well, there is an additional burden with swelling in the feet, being able to, not walk around, being completely non-ambulatory, having respiratory issues, breathlessness, all of that add to total cost of care or total cost of burden to the families, and then they kind of begin to see it. So it's also not just, so that's maybe my parting thing, is it's not just the data that you capture and you negotiate with FDA, it's also how you present it to payers is equally important.
Yeah, and I just to be provocative, because it's early in the morning, I'll get you guys worked up, is I would almost put the question back to you, because I spent a lot of time doing performance-based, value-based alternative contracting deals. I've yet to find a manufacturer that's going to put money on the table based on the type of metric that you just described. I'm not saying it's incredibly important, but is it something that you're going to put financial stake in, right? And so the payer is that a metric that you're going to use for coverage? And I would say to the manufacturers, is that a metric you want to put on the table to be at risk, right? And I don't think in today's environment we have good enough metrics in those areas that people trust systematically that they'll do it, right?
My perspective is part of a comprehensive plan. And it may not only
Yes.
be to,
Yes, absolutely.
it may not only be
Yeah.
Part
to
of the
the
picture.
payer that it matters. It may matter more to physicians and patients.
Right.
The other issue that I think is a really fundamental question mark challenge here when it comes to patient-centric scenarios, and I agree with Francesca that you have to start early. Talk to the patients and the docs, figure out what's going on, pressure test it with payers to understand how sticky it is, or if they even understand, is this something that helps make sense of what might be a surrogate? Is it something, if you look at Jakafi, the spleen volume reduction cancer therapy, it was the patient-centric data that helped make sense of what does spleen volume reduction mean. Payers were like, I have no idea, but it was the patient-centric data that carried that story for that product, so it can depend. And the other thing is our existing instruments in regenerative and advanced therapies may never have been built to measure the patient-centric impacts that we may see with some of these therapies. So thinking about like looking at Luxturna with the whole maze process and other stuff, you have to be willing to take a step outside if the existing instruments just don't fit. But we have like 1 minute, so in one sentence or a couple words, if there's anything that you would want to impart to these guys about prepping for real-world evidence, then now's the time.
I'll just say that start early. It's never this one, so start early, start thinking broad, always work with the end in mind. That is, you're sitting in front of a HTA committee, you need to justify why they need to pay the price, so start thinking that way. And real-world evidence is not a luxury anymore, at least for rare diseases, it is a necessity. So starting early is my advice.
And I would say the same. Get the payers in the room with your scientists, even prior to phase 1. Let the folks who get very excited about mechanism of actions and surrogate markers listen to who's actually paying for those medicines and what they care about.
Great. And in addition to those points, I agree. I also think there's alignment within your company that's important too, because I think times are changing in terms of what's going to be needed to support ultimately market access and payers down the line. And I think the clinical and other teams need to understand that early and sort of working really closely with you on the sort of the market access side to incorporate that real evidence into the clinical development program.
And I agree with all of that. I would just add on top of that I think real-world evidence is going to be absolutely critical for coverage and reimbursement in this space. And the more actionable that real-world evidence is in the post-market setting will be very helpful to help ensure sustainable access and coverage.
And my parting thought is we're well beyond the time when the outcome of your clinical trial is the only thing that's going to carry your product, particularly in this space. And you have to, from the very beginning, think about your comprehensive strategy because you're building a package, a story, and that story won't be only answered by your trial. It's going to have other bits of information, especially in this space, to help carry the day. But we're at time. And thank you so much for participating, and I hope you enjoyed it.